Okay, thank you. I'm going to talk about bird ears and hearing, and aquatic birds in particular. So let me give you some background about bird hearing. Roughly speaking, birds hear in a similar hearing range to humans. Just to show you here some audiograms of birds in gray compared to humans in red. And the bird ear is a tympanic middle ear, so they have an eardrum. Birds, but in contrast to mammals, they have a single ear bone. That's the columella here in blue, connected to the eardrum by extracolumella cartilage in pink. So this ear functions well in air, and we know that because birds communicate acoustically, we can hear them. But what about for aquatic birds that spend some time underwater? The acoustic medium is different underwater. It's a higher acoustic impedance, higher speed of sound, which can mean that there might be modifications to the ear to hear underwater or to protect the ear from the high hydrostatic pressures. So normally in air, we consider the middle ear an impedance transformer. So essentially what's meant by that is the pressure is increased from the eardrum to the footplate to overcome the reflection that would otherwise occur. So we were interested in examining what is the situation of the ear anatomy in aquatic birds. Have they changed some of the auditory traits that would be normally related to impedance matching for functioning in air? So we used a comparative approach to examine this question. Just to give you some more detail of some of the anatomical structures we thought may differ. So firstly, related to impedance matching, we measured the ratio between the area of the eardrum and the footplate of the columella. We suspected that may be reduced in aquatic species. We also measured the height of the conical protrusion of the eardrum, which is related to a catenary lever mechanism that increases the pressure on the inner ear, suspecting it might be flatter in aquatic birds. And we also measured the length of the extrastapedius of the extracolumella and the size of the round window, and also the offset of the columella from the center of the eardrum, which we expected may be a proxy for the lever related to the fulcrum at the perimeter of the eardrum. In addition to impedance matching traits, we also were wondering if the columella may be larger, because if we compare to pinnipeds, pinnipeds all have larger ear bones relative to terrestrial species. We also suspected that diving birds may have less air behind the eardrum, because generally they have less pneumatized skeletons, and so we also wondered whether that would apply to the head, which would have implications for ear stiffness potentially, and that also may affect the amount of connectivity between the ears, which we know is important for the binaural cues available to birds. Also, we measured the size of the cochlear aqueduct, which is a backdoor sort of opening of communicating the perilymph between the inner ear and the cranial cavity. It was previously noted that this was larger in aquatic birds, and there are some hypotheses that this opening may be related to a bone conduction hearing mechanism. So the general methods flow was we collected a large diversity of heads of naturally deceased birds through a network of our researchers and collaborators here in South Africa, and also collaborators in UK, and we got some birds from subantarctic islands as well. We did the micro CT scans, took measurements, and then we compared for differences in ear structures and related them to lifestyle, so whether they were terrestrial versus aquatic, and then further we distinguished aquatic species by whether they were just at the surface, whether they were plunge diving species, and the depth to which they dove. And then we examined PCA and phylogenetic regressions. So I'll share the PCA results here. Many of the traits that we measured loaded together on the first principal component. And this first principal component was also the major one that distinguished aquatic and terrestrial species, which I'll show in a moment. So the height of the eardrum, that's the conical protrusion of the eardrum, the size of the footplate of the columella, the length of the extrastapedius, the size of the area of the round window, the distance from the columella tip to the center of the eardrum, the size of the eardrum, and then the ratio between the eardrum and the footplate was not on PC1, but I'll show you some more details in a couple of slides on that. And we can see that here's the data mapped onto PC1 and PC2 according to different ecological groupings. So green would be terrestrial, gray surface foraging species that don't dive significantly, black plunge diving species, blue species that actively pursue prey underwater. And what's interesting is that at the extremes of this PC1, you have owls, which we know are highly specialized in terms of their auditory function. And on the other end, you have cormorants. And we know that cormorants are quite, at least one species from this genus, Velocrocorax, had a rather sensitive audiogram. So there's some connections there with known hearing abilities. Here's the same PCA, but it's grouped by the taxonomic groups. So we have, again, green terrestrial species in green, and then different orders that have at least one species that has underwater pursuit behavior. And I'm just highlighting here in black Charadriiform order, which is ecologically diverse. And you can see that it has terrestrial members which are spread to the one side of PC1, and then the diving species such as auks and puffins are further on PC1. Just to show you some examples here of the difference in footplate, we're comparing a cormorant here with the size of a turtle dove. Differences in the eardrum size within a terrestrial and a diving Charadriiforms. And then to show you a comparison between barn owl and African darter, you can see differences in the conical protrusion of the eardrum in a slice and in a 3D rendering, and also the columella offset and extrastapedius length difference. Just to show a little more detail on the tympanic membrane area and footplate area, those were lower in the underwater pursuit species, but there was the slope of that relationship between head mass and eardrum area is a little lower in underwater pursuit species. And then if you examine the ratio between them, there's a lot of scatter. But the area ratio was related to dive depth. And so it was species such as penguins which had the very low area ratio. This bottom plot is just showing a relative measure of the auditory metric as a function of head size. In terms of air behind the eardrum, the underwater pursuit species had lower cranial air, And we confirmed other observations that penguins don't have an interoral canal. And also, in a very deep diving cormorant, the South Georgia shag, we also don't see any opening in the bone. And so there's a continuum of connectivity, and then terrestrial species have much more connection between the ears. We did find a larger cochlear aqueduct in general in the aquatic bird clade. Just to show you here in a scan, this is a very large opening. It's kind of a back door to the ear. Also, we didn't find any differences in the size of the columella in terms of the volume. Some of the larger surface foraging species had slightly larger or longer columellas. There are some clear differences in morphology, but we didn't see those differences in volume. So in summary, there were several differences between aquatic and terrestrial birds. These were significantly these were related to less impedance transformation, which may explain some of the poor hearing in diving birds, although some diving birds do hear well. So we can conclude that underwater, or that an aquatic lifestyle is significant for affecting ear anatomy. This could be related to underwater hearing, but could also be protecting the ear, since some traits were related with dive depth. So now we have a nice anatomical data set to inform future research in this area. And so I'd like to thank many people who helped us, our tech Jacques Schiepers, collaborators in the Netherlands, scanning facility, and our funding. Thank you.